So I'm happy to be able to introduce Jake Yates here from Berkeley. Um, Jake and I met when I was postdocing in Austin, and he was a graduate student there. But it's probably the case that we actually, so that's the one we met and had name recognition and formed memories of one another. But it's almost certain that when I was a grad student at Berkeley, he was working the bar and had served me more than a couple of pitchers of beer, right? Because I used to frequent the place that you tended to bar after going to the gym, playing basketball in the afternoons or the evenings. So Jake is I guess I would describe it as a Swiss Army knife of a researcher. He's done, he's worked on a huge number of really interesting projects with an array of accomplished mentors. He's worked on decision making and motion processing with Alice Huck and Jonathan Pillow. He started doing some really high fidelity eye tracking during his time at Rochester, where he was hooked up with Greg DeAngelis, Caleb Rucci, and Jude, well, I mentioned his last name, Jude Mitchell, working in a marmoset. He's worked on different organisms, humans, macaques, marmosets, and then he followed up his time postdoc number one at Rochester, moving down to the University of Maryland, where he boned up on his already considerable computational chops, where he spent time with Dan Butts, and that led him into an assistant professorship at Berkeley, where I'm sure he'll soon be transitioning to associate. Um, and more than that, I'm just very pleased to have him because he's a good friend of mine and a wonderful science buddy to the top shop with. So he's got a lot of interesting work here that I think originated with your time in Rochester, right? And probably touching a little bit on stuff that even started back at UT. And he's well, you'll see. It's a technical tour de force with some really interesting conclusions that make you rethink what is, I think, has become a consensus view in this area. So let's all join me in welcoming him.
Okay, thank you, Johannes, for the very nice introduction. I want to start by just inviting you all to interrupt me at any point. This will be more fun if you have any questions or even thoughts. And yeah, I've worked on a ton of different model systems, all of them related to visual perception. And I want to start this talk by anchoring to two really long-standing approaches in understanding visual perception. The first is the idea that perception is a process of unconscious inference for causes of the sense data. This is widely attributed to Helmholtz, but others have said it even before him. And the other is that perception is neuron activity, patterns of neural activity in some select group of neurons that are representing things, which is, I'm attributing it to Barlow here, but obviously these are both very well-developed, long-standing lines of work. And I'm saying both of them, this issue of neural variability becomes a part of the puzzle. And so I want to start off by saying what I mean by that, and then I'll unpack it some more. So what do I mean when I say neurons are variable? I mean that when you put a stimulus in a receptive field, say here of a fixating macaque, and record from the action potentials of a neuron in visual cortex, the pattern, the spike trains that you get on each presentation are different. So each tick mark here is a action potential, each row is a different presentation, and each row doesn't look the same. And if you count them all up, they're variability. We've also known for a long time that if you record more than one neuron at a time, that the spike counts are correlated across the neurons to repeat perceptions. There's now over 50 years of research on this phenomena and hundreds of papers linking it to the information in population codes, perceptual states, attention, and now ongoing brain dynamics. And what I want to try to convince you today is that a large portion of this can be linked to the active sensing strategies of fixation line movements during vision, even in these constrained tasks. So the outline is really that neural variability and eye movements are two sides of the same coin in visual cortex. And I want to start, I've organized in three parts. The first part I'm provocatively calling a law of vision. Eye movements, a particular pattern of eye movements is a law of all vision systems, biological vision systems. And then I will talk about the results from the experiments we've done. Okay, so if you look at kind of natural visual experience for humans, right, this is somewhat what it feels like. There's a cyclopean view that's egocentric. Uh, Most of the transformations that are going on in the world are done by us. The world is mostly still. Of course, we're also moving our eyes. So there's typically a pointer representing where we're looking while doing these tasks. But the input the brain gets isn't this cyclopean view. It's shifting with the retina, with every single one of those eye movements, and it's foveated, not to mention that we have two eyes. So I want to start with this picture here because even though this is not an accurate modeling, I've just done a log polar transform on this on the video. It represents kind of how jarring the natural visual experience is that the visual system has to learn to contend with. And I really want to make a few points about this. First one is that the largest source of variance in luminance is self-generated transformation. The world is mostly still, and almost all of the variance is coming from eye and head movements and actions with our hands following behind that. Although a lot of the important things are stuff that's out in the world. The second thing is that the visual signal comes in abrupt jumps, called saccades, interspersed with gaze stabilization, stations, and the uneven sampling is produced. The foveation part is pretty special. So among mammals, this is only present in haploid primates, showing them representative ones on the bottom there. A massive increased density of cone photoreceptors at the center of the visual axis. Looking more broadly across the animal kingdom, foveation is also present in birds and prey. But there's uneven sampling in lots of species. But more interestingly is that saccade fixate, this pattern of correcting for head movements to stabilize gaze so that the retinal input is relatively stable for periods and then jumps, is ubiquitous. So every single biological visual system that we know of samples the world with a similar pattern. Even animals that don't move their eyes in direct saccade fixate patterns will have a gaze stabilization interrupted by jumps of the retina. So from the point of view of the retina and what the brain gets, saccade fixate is ubiquitous. I feel like this is something that I didn't appreciate until recently, just how widespread it is. Before there were rings on Saturn, before there were lungs, before there were jaws on animal, there were animals saccading fixating on Earth. It evolved multiple times, and every single species that has been tested this way makes ballistic eye movements in response to sensory stimuli, including lamprey, right? So like totally alien from humans will respond to a visual flash by moving their gaze towards it. So I was trying to be provocative, but I also think that this is a process that the visual system always expects to be going on all the time, and that you can clamp it during instructed fixation, but you can't eliminate it. And it's still the pattern that the visual system expects. Which brings me to the small scale, right? We now have very, very clear evidence that even during fine fixation tasks, the eyes are still moving. So this is a video from an adaptive optics scanning laser ophthalmoscope from Austin Norda's lab, showing a cone mosaic of a human retina during an E optotype acuity task. And you can see that the retina is drifting over the E. The flicker, I think, is noise from the scanning laser, but the drifts are quite real. And these eye movements have become a kind of a popular area of research in terms of their function of vision for high acuity tasks. So we know they're there. I also want to emphasize that you can see your own fixation lines, right? Which we could probably stop the talk here because this means propagating all the way through your visual system to things that you have perceptual access to, even though you're not aware of them most of the time. So again, there's been this recent surge given the new technology with adaptive optics in measuring and describing the function of tiny eye movements. Most of the papers start with titles or sentences like, even during fixation, our eyes are constantly in motion. And you might think that these are new observations, but if you look through the literature, you can find almost identical sentences. In ordinary use, the eyes are never entirely at rest, but are continually moving in the 1930s. So there is a parallel history to this neural variability history. That is the characterization and functional interpretation of fixational eye movements. And I want to highlight two eras here which are kind of these weird dark ages, where for a few decades, fixational eye movements were considered noise with respect to visual function. And even in the early 2000s, when people started linking neural activity, measuring responses of neurons in response to things like microsaccades, and they found everywhere they stuck an electrode that there was a modulation of firing. It wasn't cleanly linked to function. Okay, so that's what we're going to try to do today, is link this cleanly to kind of concepts of the coding variability primary visual cortex. And to do this, this requires at least two technical hurdles. The first is large simultaneous recordings of large populations of neurons, which has become available with silicon electrode arrays. We can regularly record from roughly 100 neurons simultaneously, I think, with some isolation quality. And the other is highly accurate eye tracking. Yeah.
Okay. There's a comment from the Zoom audience saying that they can only see the room.
Oh,
can you try sharing screen if Jessica set you up that way?
He did not set me up that way. Let me see if there's I was going to ask her, is there a Zoom that I can join?
I'll holler at Jessica and see if
That's not right.
just continue. I'll write her and see if she.
I found a Zoom link in an email from her. I'm going. Is that working for them?
Continue to share it. So why don't you do your best? I'll write them and find out. But
okay, great.
We should just pause indefinitely.
It says my screen sharing is paused. But anyway, I'm going to continue. And apologies to those of you on Zoom. Okay, so yeah, this requires that you can record lots of neurons and measure where the eyes are pointed very well. I've already hinted that we can measure the eyes quite well with adaptive optics. The issue here is that these are prohibitively expensive and cumbersome systems to work with monkeys. There's heroic work from Lawrence Sinsich. You can make this work in anesthetized monkeys, but there's a limited field of view, so it's not going to work for kind of natural eye movements. The next best case, I think, is dual Purkinje. These were also historically very cumbersome systems, analog servomotors tracking with photocells. But in recent years, there's been an emergence of digital solutions. Point a camera at the eye and measure the reflections off the front of the cornea and the back of the lens. And I want to show you this one figure from our video from Tabanero et al. Showing how these two move together. So you'll see that the little dot in the back there is tracking the back of the lens. During a saccade, moves differently than the reflection on the cornea. The difference between these two reflections can be used to infer the angle of the orbit. So in my lab and in collaboration with Jorge Otero Mion, we have made an open source version of this, OpenIris DPI. I want to make a quick pitch for it. You can build it. It's inexpensive. My student Ryan has gone to tremendous effort to make a very nice wiki to explain all the principles and how you build it, complete with step-by-step instructions. And then when you use it, it's a nice GUI, so it's not too bespoke. And it's much, much cheaper than an iLink. So those of you that use iLinks, it's much cheaper and it's also much better. Once you have this set up, you can simultaneously measure the pupil and the dual Purkinje signal. And they broadly agree. So as a subject looks around the natural image, showing here the vertical and the horizontal eye trace, there are general agreement between the pupil track and dual Purkinje, but they don't entirely agree. And in fact, you can see during fixations, the red traces will show up below or above the black trace, which is the DPI. And zooming in, this can be roughly on the order of magnitude of about 5 to 10 arcs is the disagreement during fixation. So which one of these agrees with the retina more? We did a study at Berkeley where I would say, Austin and an optical engineer built a retinoscope that can track both eyes simultaneously with enough space in the optical path for one of these video eye trackers. The answer is that during fixation, the DPI signal is much more correlated with the retinal signal than the pupil is. We've also confirmed similar things, trying to correct for receptive fields in macaque LGN that suggests that basically we're getting not just a different signal, but a better signal for where the is. So going back to this, like, of course, people have conditioned on fixational eye movements before, right? Like no neurophysiology paper on neural variability and attention or internal state has not checked for fixational eye movements. But they've all done so with either pupil trackers or eye coils. And eye coils can be quite accurate, but they're difficult to calibrate well. And so there's lots of reasons to think that that signal, especially in neurophysiology rigs, wasn't the focus of the experiment. So it leaves open the possibility that there's a strong connection. All right, so that's the project that we set out to do. I want to highlight the people doing the work. The grad students in my lab, Ryan Ressmeyer and Tejas Kotapalli. Our chats. Are the chats saying that they can see this? Or
I haven't gotten anything else.
Okay, cool. All right. And then Declan Rowley's a postdoc at Alex Hock, who have been collecting more data with simultaneous pupil and binocular tracking. Unfortunately, so what we do is we have marmoset monkeys fixate on the center of the screen. They have to maintain their gaze in a fixation window, and we show rapidly flashed filtered natural images at fixation. So they're able to actually move their eyes around on the stimulus that they're fixating on. But the window is forcing them to be constrained within a 1 degree radius, and they tend to keep their eyes well within. So that's the distribution of eye positions across an experiment. We're recording from neurons in the foveal representation of V1 at the very center of gaze, in part because now they're covering our stimulus. Also, this is where we expect there to be, I think, most of the consequence of fixational eye movements are for things that you're looking at. And it's massively overrepresented in V1. So we all know that the visual field is mapped onto cortex in what is known as retinotopy. This central 0.01% of the retina, which is the foveola, this very central part we're recording from, maps onto about 8%. So when we record from these neurons, look at a single trial, we can see the pattern of activity across a population. Each row now is a neuron, not a trial. So this is one trial of activity. And what I want you to notice is that over these 500 billion milliseconds, the neural activity kind of moves up and down together. And if we look across multiple trials, we can see that this pattern of activity changes. But so the two things are the population tends to move up and down together, and that different trials have different patterns. Now, if I go through the data and I cherry pick trials based on the similarity of their eye position during an epoch, what I'm showing you in the middle here is, so the monkeys make microseconds just like humans, but there will be periods of relative fixation. And I've picked red trials and blue trials where the distance between these is about 0.15 degrees between the two conditions. And then I'm showing on the right the spike rasters from the red trials and the blue trials. And then I'm going to play through this movie, and you can see exactly how close these eye positions are. They'll be, yoked right next to a feature of the image, but produce wildly different patterns of responses across the house.
Red's on the bottom, blue's on the top.
Red is on the bottom, blue is on the top.
What's the basis of dividing up the red and the blue? How are those selected out?
This was basically clustered based on distance in the fixation on the screen. Yeah, and if we wanted to analyze things the way people have in the past, where we would say, I'm only going to find all the times where the eye trajectory was very similar, and so then I'm going to be able to then look at the variability around it, we end up with no data, right? So it's very hard to find conditions where the stimulus was exactly the same and the eye trajectory was exactly the same. So we're going to need a better approach if we're going to assess how much variability is due to fixation line. We're going to need to do a little bit of math to figure out how to do this. So the approach we're going to take is to apply the law of total covariance, and I'm going to walk through this first in with the equations, and then I'm going to walk through with pictures. So if you don't like equations, you can wait till you can listen to the words and wait till the pictures come. But basically, we take all of these trials, we lump them together, and we calculate the covariance over trials and time. Right, so this is the total covariance, which is going to be a neuron by neuron matrix. The diagonal of this matrix is the individual variability of the neurons, including all signal and noise. And the off diagonals are the shared variability. The law of total covariance says we can factorize a covariance matrix like this into an expectation of a conditional covariance, a covariance of a conditional expectation. All right, what does that mean? So the first term here, t is time, and e is the eye position, or eye trajectory. It's a 50 millisecond trajectory. And I is trials. So the term on the right here is the rate variability, saying if I condition on time, well, that means that the stimulus is exactly the same because I've showed repeats of the exact same thing. And I condition on the eye position, that means the eye trajectory was exactly the same. If I take the expectation and average across all the spikes, that's the rate. So this term on the right is the covariance of the rates. And the other term is what's left. It's the variability around that. So this is the internal noise and the rate variability, and these covariances have to sum. We can apply the same trick again on the rate variance and factorize it into the expectation of the rates conditioned on only time. So averaging over eye movements. Looking at the covariance of that, that's the covariance of the PSTHs of the neuron, the covariance of the average rates. And the first term is what remains is the fixational line movement covariance. This is the variability that is due to changes. And these things all have to sum. So we end up with basically 4 covariances, 3 of which have to sum to equal the other one. And we're able to estimate from data 3 of these. So we can measure the total covariance, we can measure the PSTH covariance, and we can measure the rate covariance by conditioning on pairs of trials where we are pairs of trials and times where the eye trajectory was the same and the stimulus was the same. And we can build up a big bag of these pairs of spikes to estimate the rate. And then the fixational eye movement covariance is just the subtraction of that rate covariance and the PSTH covariance. Okay, so what does this look like in pictures? Here's a neuron. That's its mean PSTH on the bottom. It's quite variable. There's some standard deviation is large around that. And if we look at different trials, we can see that the eye trajectory or eye position was slightly different on these 2 trials. The spikes were also different. And what we're going to do then, when we build this big bag of spike pairs, is to take the times when the trajectory was similar in a 50 millisecond window, and we'll take the pair of spikes from 2 separate trials and put them into a bag that is linked to a bigger difference. Sorry, let me say this again. I got distracted by the idea here is that we can take as long as the time in the trial is the same and the eye position trajectory has a particular distance, we can put that in a distance bag, right? And we can do this for different distances. And what this lets us do is build up a distribution of all possible pairs of spikes that have matched stimulus and trajectories, right? And what this has done is change our conditioning down to having only 4 trials left where they are the same, to having 4,000 pairs of spikes from different trials where the eye trajectory and stimulus are the same in the smallest bin. And for many of these other bins, we have many, many more samples. So then we can estimate the covariance in these different time points. So let's say look at the red covariance, and we'll get some, spikes squared per second is the covariance in this eye trajectory distance. And if we look at the yellow group, it's a smaller number, and at every single bin in between it falls off. So what this is saying is that when the eye trajectory distance was quite small, the neuron was highly reliable, right? It was strongly correlated with itself across trials. So by using this approach, we're able to increase the statistical power to factorize these covariance matrices and take the total covariance, which again was made up of the individual variability and the shared variability, and break it into what can be explained by the PSTH, right, which has some of that shared variability structure, but not much of it, and what can be explained by fixational line movements, which, judging by the color map here, is a brighter color map. We're going to quantify this next slide. And what is left is the internal noise, which looks mostly diagonal, with a little bit of negative log dynamics. So this is the approach that we use to basically factorize out these different sources of variance. And then we can quantify across all of the sessions and across all the neurons how these relative magnitudes are distributed. So the first thing we can ask is, across all the neurons, are the fixational line movements or the PSTH driving more of the rate variability? And that's basically what the magnitude is along the diagonals here. If the number is greater than 0.5, then fixational line movements are explaining more variability than the PSTH is in the fluctuations of rate. And across the population, the median is 0.8. So roughly 80% of the rate modulation is linked to fixational line movements. Yeah.
Sorry, maybe I totally missed it. In there, were you accounting for any lag between the eye movement and when the spike is occurring? And how do you account for that?
Yeah, so the trajectory similarity is conditioned over a 50 millisecond history. And then I count forward in time the spike count. And the trajectories have to be the same for the entire window that I'm counting over. You're going to see this limits how much time we can count over. So basically, we're going to have really good estimates of the contribution of eye movements to spike variability in like 10 millisecond and 20 millisecond windows, even 40. And it starts to break down by the time I get to 80 milliseconds.
And can you just say real quick, what's roughly the size of the receptive field of these neurons that you're measuring relative to the size of these fixational eye movements and the distribution?
Yeah, that's a great question. I don't know if I touch on this directly, but the distribution of, for many of these neurons, the distribution of fixation line movements, it expands a little bit bigger than some of the receptive fields, right? I'll talk a little bit about this, but that's not the entire story. It turns out it's like sensitivity to phase and small shifts of the image is present across the entire population to some degree. And so we see subreceptive field shifts matter. But that is a good question. I'm going to try to anchor this with respect to size of receptive fields more broadly. You're right, we're in the fovea. This is like where it should matter the most.
Have you put effort into also typing the neurons into simple and complex cells?
Yes.
Yes.
I have. We find that roughly 80% of the cells in marmoset B1 are complex by conventional measures. That doesn't mean that they're not sensitive to shifts when there's phase alignment, but on gradings, they're pretty phase aligned. Okay, so we can also use this to calculate conventional measures of neural variability like nanofactors, just so we can see where we're sitting. So it's possible that our receptive fields are so small and the eye movements are so big that we're actually driving much higher variability than normal, and we're only explaining away some of it. But when we don't correct, we still get a kind of typical Fano factor of around 1 in these neurons. And when we do account for fixation line movements, it now drops down below 1, about 0.77. So it's explained away some of the boson-like variability that is present. And this is robust across counting window sizes that we have enough statistical power for. So as you increase the counting window, the Fano factors grow in the uncorrected case, suggesting that the eye movements are actually driving a lot of variability by moving the receptive field around. But they stay low for the windows that we have statistical power for. We can do the same trick for the off-diagonal terms and calculate the noise correlations, which are typically around 0.05 or 0.1 in B1. And we find that as well when we measure uncorrected. And once we correct, it drops to about 0. And this is also robust across counting movements. So having corrected for fixation line movements, we've mostly removed the correlated structure across neurons. What's left is actually a small amount of anti-correlation and imbalance. We can further ask questions about the dimensionality of these signals. right? Because there's a lot of discussion about shared variability, being low dimensional, in reflecting kind of internal state, things like that. It's often characterized as a low dimensional gain. So if we calculate the eigenvalues and eigenvectors of the covariance matrices here, well, one, we can see that the magnitudes are slightly larger for the eye movement covariance than for the PSDH. That's what I already told you about their influence on the rate variability. But if we calculate the effective dimensionality using the participation ratio, the fixational eye movement covariance stays pegged at 2, no matter what the PSDH covariance dimensionality is, which well matches the dimensionality of the signal that we fed into conditional, right? So we're measuring monocular eye trace in this case and conditioning on that. Now, these are 2 different covariance matrices. We can ask whether they're aligned with each other, right? Each one of these looks like a pattern of activity. There's some hints that these patterns are similar, but across all of the different sessions, how similar are they? There's lots of ideas about whether shared variability is information limiting, which would be the case where it is moving variance along the stimulus selectivity, or whether neural variability is orthogonal to that, and that doesn't matter. So each of these covariance matrices is an ellipsoid in the n-dimensional space of the number of neurons that you have. And the principal components then represent the axes of variance, orthogonal axes of variance of that ellipsoid. And so we have 2 covariances and 2 shapes. And what we know that our fixational eye movement covariance is roughly always 2D, it's dominated by 2 dimensions. But what we can do is we can ask, how much do the eigenvectors of the PSDH covariance explain the fixational eye movement covariance? Right? And in one case, they're aligned. And that means that if I project the fixational eye movements onto the PSDH eigenvectors, the first 2 of them, then I'll capture most of the variability. And then in another case, they're orthogonal. Shifts of the retinal image are going to be consistent with them being aligned. Whereas it's possible that extraretinal input that can modulate these things in a way that is not related to their signal tuning could be orthogonal. So what we do is we take the covariance from one, project it onto the eigenvectors of the other, and then calculate a trace ratio. What the trace tells you is the total amount of variance, the total volume of that ellipsoid. And what the numerator is now saying is how much of that volume is captured by the first 2 eigenvectors of the other covariance matrix. Right? And across all of our sessions, the number is quite high. So basically 75% of the total volume of the fixational eye movements covariance is captured by the first 2 eigenvectors of the PSDH covariance. So they're highly aligned. This means a couple things. It means that the effective fixational eye movements, if you didn't know what they were, would look like a low dimensional, a 2-dimensional gain in this case, because it is modulating responses along the dimensions of variance that are already present. All right, so to summarize so far, the trial-by-trial variability in V1, central V1, is very strongly linked to fixational eye movements. It drives roughly 80% of the rate modulation. Conventional measures of neural variability are reduced after correct conditioning on it. And the fixational eye movement covariance is 2-dimensional and lives in a subspace that is largely defined by the PSDH. I think this is a lower bound for a number of reasons. This is tracking one eye. We're working on getting the binocular setup data up to speed. But there are also vergence movements that are happening on a scale that will move eye movements, move these receptor fields around. A lot of these neurons are binocular, and I don't know what the dominant signal is from the 2 eyes. So this is, I think, a decent approximation for the contribution of vertigo on neural variability, but not vergence. There are also other eye-dependent changes in the retinal image that could drive variability. The bigger question then is, how is this changing what we think about what V1 is doing? How does this change the encoding process? All right. Any questions before I go on to that section? Yeah.
So the matrix you showed, it's not uniform. There are parts that are dark, parts are
right.
What's different between them?
So each one of those indices is a pair of neurons, right? And when it's dark, it means that those neurons really co-fluctuate together.
Right, like what's different?
What's different? So what's different is I think largely that, so these recordings were made with two neighboring silicon electrode arrays that were separated by 200 microns. And so we're usually in two columns, right? And the neurons in one column will move together quite a bit. And the neurons in the neighboring column will not necessarily with them exactly the same. So that's what I think is driving that, but I have not pulled that apart at all. Okay, so how do fixation line movements modify encoding? To approach this question, at minimum, we need an encoding model. This will both let us disentangle feed-forward and extraretinal contributions, but also see how well we're doing at using things like the retinal image and fixation line movements to even explain the spiking variability. And to do that, we're using a digital twin approach, where we train a deep neural network to predict the spiking activity of the neurons by minimizing the Poisson loss on spike counts in 10 millisecond bits. The model in question is a very large model by neuroscience standards, but a very small model by machine learning standards. It's got 4 million parameters. There's I think 2 million of them are in what is called the vision core, which is a series of temporal filters, so temporal only, and then a 3D spatiotemporal ResNet, which is just a big nonlinear feature extractor that trains nicely, and then a convolutional gated recurrent unit, which is a recurrent neural network. Why not something that's actually matched to the visual system? Because I spent years trying to make that, and this trains more robustly across all settings without lots of hyperparameter tuning. And they're not interpretable anyway. So at this point, we just want to know if we condition on the retinal input, how well can we explain spikes? We also, for completeness, include an optional modulator network where we pass in the recent eye trajectory history and let it modulate the output of the network by concatenating the activations to the output of the ResNet before going into the RNN. So the RNN has to learn how this thing's. The output of this core network is convolutional. So it has a response of a feature vector of channels for every single position in space. And we map to the spiking activity of individual neurons measured across sessions with a spatial location in the convolutional output and a feature vector across the channels of the network. And what this lets us do is combine across the 20 recorded sessions we have to train all of the parameters of the core are shared. So all gradients of this loss, every single sample contributes to the core, but the individual neurons have their own mapping. And we train this entirely on free viewing data, where the monkeys are free viewing natural images and also artificial flashed noise stimuli. And so I just want to show you a kind of picture of an example image that was withheld from the training set. What you can see is the marmoset is looking around. The input to the model is shown in red. We do a lot of work to calibrate the eye tracker, make sure that we're reconstructing the exact position of gaze on the image. I've swept a lot of those details under the rug, but this is a picture of what the model gets in the bottom left.
Just to be sure I'm not misunderstanding here, this is the picture that another marmoset is staring at.
Yes,
right?
this is
This
this picture is displayed on
a
this picture is displayed on a computer screen. Marmoset is head fixed looking at it with an eye tracker.
And what the marmoset is looking at is a picture of other marmosets.
And the marmoset is looking at a picture of other marmosets. Yes, in this example, I like this picture because it likes to look at the faces of the marmosets. The eye position in pixels is shown on the bottom. The observed spikes are shown on the top. And now I'm going to unveil what the model prediction was to this withheld stimulus. And so broadly speaking, the patterns of activity match. So the model follows saccades. It looks like a blurred out version, right? This is a rate model that if you drew Poisson spikes from these rates, it looks a little bit more like the data. But we can evaluate how well this thing is doing using kind of more conventional measures. So if we interrogate the digital twin of a bunch of neurons and measure the receptive field of it using noise that wasn't shown during training, we find that the model has nice receptive fields. That's shown on the right. So each one of these little pairs is the data on the left and the model on the right. So when the neuron has a nice receptive field, the model has a nice receptive field. They tend to match. And we can also evaluate it on our withheld fixation condition and say, well, how well can we predict the responses to this flashed fixation condition? This is a true test of generalization because this entire condition was withheld from the training set. And here's an example neuron. And if we overlay the model prediction, it lies kind of right on top of it. What I want to highlight here, probably hard to see, but there are dashed lines around the model prediction here. Those dashed lines are 95% intervals, the quantile intervals for predictions from a model that is deterministic and only given retinal input. The reason it is variable is because the retinal input has been shifted by the animal's actual eye movements. So this is the amount of variability that a deterministic feed-forward model given realistic retinal variability would predict. If we look at the correlation between the data PSTHs and the model PSTHs across the population, we get about 0.7 on average, which is about as good as anybody does with this kind of approach. And usually they're not pushing at this time resolution. So, for some reason, this 0.7 shows up all over the place. This is what Andreas Telias's groups gets. This is what we all get. There's some additional variability that we just cannot capture all of it through these approaches. But we can now get back to our question of how much variance is due to fixation eye movements. How is this related to the other measures I showed you? So we can compare the single trial R-square, so how much of the variance on single trials are we explaining, with the retinally shifted input to the model, compared to if we just copy the PSTH over and over again, and the noise was not related to the retinal shifts. So if the model is doing better than the PSTH, the points will fall to the left. And if the model is doing worse, the points will fall to the right, and they mostly fall to the left. The model is doing better than the kind of conventional best you could do is the PSTH. Perfectly predicted it. That's the best you could do given the stimulus if you didn't take fixation line movements into account. Now there's some notable exceptions to this where there are points far on the right. When we look at those neurons' PSTHs, they seem highly entrained to the image update in our stimulus, and they're not particularly spatially selective. Finally, the relative gain in performance using the model with true retinal shifts over the PSTH is highly correlated with our other measure of whether the neuron was dependent, like how much its modulation was due to fixation line movements. Now, the fun thing about this is that we can now take this twin and do a bunch of in silico experiments, right? So we take all of those separately recorded neurons and we collapse them into one model where we can look at the convolutional output of every single twin of a neuron. So what we're going to do is we're creating virtual copies of the neurons we recorded from, and we're going to be able to watch movies of what is going on in V1 to this population of virtual neurons. So to orient you here, I'm just going to show you a small subset of those. On the left is the input to the model. It's going to be jittered with real fixation line movements. And on the right, each one of these little insets is spatial copies of one of the twins. So I've taken one of the neurons we recorded from, I've taken its digital twin, and I've made spatial copies of it, distributed so that I could see what would happen if I had many, many copies of this neurons across spatial space. This then builds up a population of neurons that is equal to the size that would actually be there in V1, which is more than we can record. So we can watch through, and again, a deterministic model, what the effect of eye movements is on the population. So every time there's a microsaccade, there's a volley, almost a traveling wave of activity that results across the different latencies of the neurons. And you can see substantial flicker that's happening just from drift, depending on which feature has moved over the receptive fields of the neurons. And we can do this for different stimuli. Here's looking at a face. And so some of the neurons are highly spatially selective. You'll see, and feature selective, you'll see this kind of like little kind of beams of activity. And some of them are blurrier, but that's all changes depending on what the eye movements are. And this is fun because we can then do counterfactual experiments. We can say, well, what happens if the model received perfectly stabilized input? So we're going to basically take video look at the pattern, this is just selecting one neuron where there's a particularly pronounced effect. But if we take the pattern that led up to this time point, just flatten the trajectory so you no longer get your drift, the stabilized response is blurred out. We can quantify this effect using the single spike information, which is measuring basically how peaky this distribution, this output is on the right. So this is asking, how did the temporal pattern of eye movement change the spatiotemporal pattern of luminance? How did this change the spatial information at this time point? And what we find is there's a strong dependence on how fast you are updating images. So if you, the experimenter, are flashing images, fixation line movements actually slightly hurt the information. But if the image is static, fixation line movements help. We can further change image content.
So just to anchor that last statement, what's the temporal rate at which you're being hurt? Like how quickly do you have to go?
The crossover point is about 4 hertz. So which is curious because it's roughly how frequently we make saccades. Anything less than that, the digital twin says fixation line movements are good. Anything faster than that, it doesn't. It also is linked to the spatial frequency content of the images. So here's an image with fairly high spatial frequency content. And if we look at the single spike spatial information, this spatial information rate over time relative to the stationary counterfactual trace, we can see that the blue is hovering above for almost all time points. Whereas when we take a lower spatial frequency image, it's particularly favored during microseconds. So there's an interaction between the content of the image and the eye movements and the encoding process. I have not, actually that hasn't shown up yet, sorry. There we go, there we go. So I haven't got a good summary for this yet, but it's hinting at stuff that Michele Rucci and Jonathan Victor have been talking about just with linear systems, that there's an interaction between the size of your fixation line movements and the modulation of spatial content. So what this suggests is basically, the twin is a big nonlinear model, fit only to predict spikes. And now we're interrogating. And it's giving us an answer that's very similar to this principled linear systems approach from Michele Rucci and colleagues, showing that how you move your eyes to drive temporal modulations. This is saying those temporal modulations then are converted back into sharper spatial representations. So the role of fixation line movements depends on what the stimulus is, both in time and in space. The digital twin approach is not largely interpretable. It's this big white box. You can open it up, you can differentiate through it, but it does let us do in silico counterfactual experiments that are hard or impossible to do in real subjects. Okay, so you might object for a couple reasons. So one, how much does this matter? We're in the fovea of V1. This is probably the place where it matters the most. The receptive fields are tiny. That question actually already came up. And receptive fields grow as you move in eccentricity, where they get bigger further away. The point I want to make is that the receptive fields are still smaller than the magnitude of fixational eye movements and likely sensitive to the phase shifts out to a region that covers half of V1, right? So half of one of the largest areas of cortex and the bottleneck for conscious perception is composed of neurons that are likely sensitive in this way to fixational eye movements. And so I would say, roughly half of the neurons in the 2 largest areas of cortex. I don't have V2 to show you right now, but I would suspect that they're operating under similar levels of sensitivity to shifts. And more than half of the ventral stream areas are selected for this region of visual space, right? So if all of this variance is present in V1, then the question of how it gets integrated out and how it becomes invariant is actually the interesting question, right? There is this robustness to integrate across the spatiotemporal pattern of spikes that are coming from a large population of neurons. So I think that, yes, the receptive fields are small, but also small receptive fields are a huge fraction of V1. What does this tell us about what V1 is doing? and how we should think about this. And I don't have a complete picture for this yet, except that I think this self-generated causal loop is going all of the time, even in the experiments where we try to eliminate it. And so I want to return to this idea of perception as inference and see whether it gives us any hints. There's a large family of models that have formalized perception as inference as Bayesian posterior inference. And a subset of those over inference over a generative model, where the brain has its own model of the input that's coming in. Probably the most famous versions of these are predictive coding and sparse coding, where there is a linear generative model of the image. Image here is X, the activity of the latents in the brain, so your V1 neurons is Z, and the image is reconstructed, right? The goal of the visual system is given an image. How do I set my latents? And it's like a messed up build on the next one, but all of these approaches lead to the same basic inference dynamics, which is a very familiar neural network algorithm, where there's a feed-forward receptive field, and importantly, competitive interactions between neurons. Right? So similarly tuned neurons should compete with each other because they both explain the same thing. Right? And what that predicted was that there would be negative noise correlations between neurons that are similarly tuned. And for the longest time, the field has consistently found the opposite effect, where noise correlations are higher for similar. In contrast, more recent work using holographic stimulation to actually perturb individual neurons and measure their influence on neighbors has found things that are consistent with this kind of theoretical prediction that there should be competitive explaining away between neurons. And I think that there's a hint here that when we corrected for fixation line movements with similarly tuned neurons coming from similar columns, that we got, oh, there it is. Yeah, sorry. That we got negative noise correlations. And it's actually quite trivial if you put any amount of fixation line movements on this very, very simple inference model, which is not a correct model of the visual system because it has no front end to it. It's just drifting an image around. You trivially drive massive amounts of correlations, even with competitive explaining away. So I think this hints that we need kind of well, one, it hints that the theory made a prediction about competitive interactions that was inconsistent with most of the measured data for a long time, that I think was a puzzle, and fixation line movements in part help resolve that puzzle, although there's a lot of work to do on exactly how they're integrated into this framework. Okay, so to summarize, the digital twin on the what is V1 doing part, the digital twin supports an interpretation that fixation line movements drive variance through shifts in the retinal input, through reafference, and that the effect depends on what the spatiotemporal frequency is of the input. So if you're in an experiment where you're flashing stimuli or things are moving really fast, then they're still there and they still drive variance, but they hurt your input. This will affect a huge fraction of neurons, and that likely has consequences for downstream areas that pull from central vision. And that I think this might help unstick us on some of this linking of theory to neural activity. So to summarize, I told you that eye movements should be thought of as a law of vision that can't really be turned off unless probably you anesthetize the brain. A large fraction of variability can be linked to fixation line movements in central V1, and this is likely due to reafference of the eye movements. And I think this should push us to be thinking about perception as a process that is operating on top of an infinite causal loop, where the sense data is extracted to form representations, which then form actions that then form more sensory input. And that perception is a kind of slower bottleneck process that is happening on top of this, opposed to the kind of more conventional sequential stack that we have on the left. Okay, I want to acknowledge all the members of the lab and funding sources, and thank you all for your attention.
So typically we think of perception in the ventral stream, which is where you focus, but most of the action we think of in the dorsal stream, where the receptive fields are larger, and I'm trying to reconcile those two kind of competing views of in that last slide in particular.
Yeah, so my view would be that the ventral stream stands particularly well to benefit from these fixation line movements. I think it's the place where they're going to be the most important part of the code. For the dorsal stream stuff, these are typically larger scale, motion selective things that give you information about the 3D structure of the world and how to act in it. The fixation line movements are when you have stabilized a lot of that. And so the place we're talking about, there's small amounts of retinal motion, very small amounts of retinal motion. And the ventral stream has a large fraction of neurons that are selective to slow speeds. And that this also hasn't fully been explored because it hasn't been a kind of target of the conventional way of thinking about ventral stream neurons. That's my thinking.
And so just from a perceptual standpoint, how would I measure this? The prediction is I could recognize a face better, for instance, with more fixation line movements. And what would that causal or how would I-
Yeah, I mean, so the thing is, it gets complicated to do because, like, in a typical experiment, when you flash on a face, right, the transient from having onset that face is actually giving you spatiotemporal information that your visual system is quite responsive to. It's kind of the peak sensitivity is around 7 hertz, and there's going to be a strong component of that in that flashy face. Similarly, saccading to a face is going to drive a transient with slightly different spatiotemporal statistics. What it means is the representations are formed to be stable across spatiotemporal syndrome. I think it's time to start, like, we've had this wonderful success by showing images to CNNs and predicting spikes to flash stimuli. But that's not a condition that the brain ever received. It really did receive ongoing spatiotemporal stimuli most of the time. So I don't know exactly how you would test this perceptually. Like, what are the big consequences of this? Probably not that big for summarizing your ability to discern. But for sure, if you're trying to figure out if it's your friend far away, you're going to move your eyes around on the face. Yeah, Josh.
First of all, it's fantastic. You know you're old when you see someone give a talk on their well-established research program, and that was someone who invited you to give a talk. It was great. So I totally buy this idea that these are important and you know, sort of the numbers line up, but I'm just wondering how much you've thought about sort of cross species, right? That quantitatively, right, like the size of the receptive field and the size and frequency of these fixation line movements are going to make a big difference in terms of their contribution to the overall related variability. And so, like, your idea, it's about half.
And you're thinking, like, what about in a mouse?
What in a mouse? Oh, well, yeah, there's not a mouse, but a monkey.
Right. So the marmoset is an interesting case because its circuitry scales. The main difference in the foveal sampling is actually just eye size, right? So they have about half the acuity. The receptive fields are bigger, but entirely by the fact that their eye is half the axial length. And so I think that this gets effect will be bigger in, say, a human or a macaque. I have thought a bit about the mouth. There's some real puzzles in that literature because that they've quantified mouse optics quite well, and they're not total garbage. They're actually really good for things that are close up, and they cut off at about 11 cycles per degree, which is well above the kind of cone-dominated mouse acuity. Mice aren't cone-dominated visual systems; they're rod-dominated, and their rods actually tile at the right Nyquist. So it suggests that if you measured grading acuity in a mouse, you're going to get really low spatial acuity. But you could have a more active sampling strategy in a mouse that leverages patterns of activity across many rods. As long as there's nonlinear pooling, that signal can get through, and there is, that could support higher acuity vision. So I think there is puzzle for the other species as well, but I think it's the right framework from which to approach vision.
I've got one.
Yeah.
So nice job demonstrating that a good bit of the cortical variability is driven by these fixation line movements. You might not be surprised to hear me ask whether you've put effort into trying to recover exactly what the eye trace signal is from neural activity itself. Because it's a hell of a lot easier to use the image to estimate that sort of thing, or at least in principle you would think it would be, than having to read out extra retinal signals and integrate those systems directly. So I'm wondering what fidelity on the reconstruction of the eye movement trace if you've done it, you're able to get by analyzing the neural activity itself.
Okay, so the answer is it depends
Yeah,
on the image, and what neurons you're recording.
Right.
So there's two ways to do this. One is to take the encoding model and ask basically whether different shifts generate better predictions of the sites. That's an approach that Dan Douglas has used, and it's called model-based eye tracking. And if you assume a lot about the way the eyes move, like that they really have to be smooth, then you can get back better, accurate positions. You don't get the dynamics of the eyes.
Right.
We've also, my grad student just said, how well can we decode eye position using a transformer that is trained on all of these data? And surprisingly well was the answer. But I also told them don't do that. So maybe we'll pull on that. Yeah.
Your neurons that weren't as sensitive to the FEMs and were better represented by your PSTHs. What do you think those are doing in the microcircuit in V1? Are they encoding something completely different? Are they different types of neurons?
I don't know. So we have all of the laminar depth information. We have all of these conventional characterizations of what these neurons tuning are. We have a subset of neurons that seem really genuinely saccade linked, like they have a saccade response that is directional, regardless of whether there's a stimulus on or not. And we have not been able to position any of this together because it's just a huge amount of alignment of tables that we're doing after we kind of finish wrapping up this story. I don't have a good answer for you. Yeah.
This is really lovely talk. You mentioned that your digital twin had a temporal front end model. Could you just say a couple words about what were the principles that guided the design of that temporal front end and what kind of effects that has upon the results of the modeling exercise?
Yeah, the temporal front end is just 4 learned temporal filters that extend 150 milliseconds into the past. This forms a basis that can be combined by the first layer of the ResNet so that they're now building these spatiotemporal filters that have a longer window without having to have tons of parameters. I had a real dream that I was going to take Frederiki's biophysical cone model and put it in the front and just get 3 wins. That didn't happen, and it was much more cumbersome to train. So I think there's a lot of work if we want to use this approach to make it more matched to biology. Like obviously, if you want to make statements about midget and parasol ganglion cells, you need those in your model, and we don't have them right now. And I'm not sure whether that complete descriptive approach. I think that's complementary to this approach. I think this is a ceiling that we can use for starters, but I've struggled to make the mechanistically interpretable models performance. So,
Thanks.
yes.
Yeah, so I mean, from what I don't study it's correlation in activity. And your data is very convincing that that movement has an effect. But from what I learned, it's like attention modulate their measure variability a lot.
Yes.
Do you also see attentional modulation of fixation of eye movement that would explain that?
So there's a cottage industry on exactly this phenomenon, which is that attention does modulate fixational eye movements. For microsaccades, the effect is clear. For drift, it is subtle and tiny, and there's one paper that suggests that maybe what you're looking for can change your drift. I'm not trying to undo, like, I believe in internal state in the brain. There are huge effects of arousal. We've known this for 100 years in terms of, put EEG electrodes in the cap and have people do mental math or stare at a blank screen. It will change the dynamics in brain. Like, so we know that there's internal things we're going on. And so I don't think that those results are necessarily undermined. But I do think that a lot of the kind of up and down state shifting that's going on could be linked to changes in retinal input that's resulting from it.
That looks like a wrap.
All right.
Thanks, everyone.